Okay, so today I'm going to present about a very recent work that I have done with my PhD student Somnath in collaboration with Harsh Soni and his PhD student at IIT Mandi. And this is about understanding the origin of run and tumble motility. So on the left side, you are looking at a microorganism called Chlamydomonas. It swims, it has like two flagella which beat like in a breaststroke fashion. And every time the flagella are synchronized, it undergoes a run-like motion. But when this flagella gets asynchronized, it tumbles. And on the right side, you see our experiments, and these are like macroscopic experiment. And remember, this is in a fluid medium. These robots are just moving on a floor, no medium at all. But I will show you how we gain some insights from these experiments, apply those ideas to these macroscopic systems, and try to uncover origin of run and tumble motility in microorganisms. Okay, so statistical physics community should not really need the intro to active matter, but very briefly, I always love to present these two movies. On the left, you see these pollen grains under a microscope, and they are just jiggling around their mean positions. And you can basically describe the mechanical state of this system just through a canonical ensemble in contact with the temperature bath. And if you look at, and if you take a large collection of such particles, say molecules or atoms, you form traditional condensed matter phases, for example, a jelly. But on the other hand, if similar particles of the same size, they have a mechanism of converting energy, or in this case, it is in contact with two chemical potentials. So it acts as a machine, okay? It's not really in equilibrium, and collection of such particles, for example, cells of jellyfish, it forms a jellyfish, right? So this is one way to set apart jelly from jellyfish, okay? So that is how I see these living systems. And one very important aspect of living system is the motility, all right? So, and this again, it's very self-explanatory. The simplest example is kinesin motor, we saw in the morning as well. It's a simple chemical reaction from ATP to ADP hydrolysis. But of course, if you move to larger and larger length scale at animal scale, this emergent property of life and free will complicates the things, right? It's difficult to explain the motion of a whale, for example. But what we are making small strides actually, and let us try to understand the next step from the motor, like microorganisms, like E. coli or Chlamydomonas, and this is what I'm going to talk about today. Okay, so run and tumble motility, it's very common in these microorganisms, and in simple words, it basically started by these very beautiful experiments done by Howard Burke many years ago. So it's an E. coli bacteria which has many hair-like active filaments, and these filaments are like doing, they are rotating clockwise, and every time one filament undergoes a counterclockwise or other way, so it undergoes a clockwise rotation, it tumbles. So it is running in straight line, and suddenly it undergoes a tumbling motion. And people have down the line started working with even simpler organisms, Chlamydomonas, which has only two flagella. And there also they saw the similar thing. This was the movie playing on the first slide. So when the flagella are beating in synchrony, it is running, but when it undergoes asynchrony, it tumbles and it changes its direction. And yeah, so that's exactly what I said. Synchrony leads to run event, asynchrony leads to a tumble event. And the current understanding essentially is this hydrodynamic coupling between these flagella. All right. So think about these. Okay, so these flagella are all in a fluid medium. They are beating and they are getting synchronized due to fluid medium. Think about these metronomes, which are on a wobbly platform. And in this case, the metronomes are the flagella, and the wobbly platform is the fluid medium around it. But also quite recently, people have observed that there are some inconsistencies in the picture of hydrodynamic synchronization. Especially, they found the role of, it's not very clear here, they found the role of a distal fiber here. It's an elastic connection between these two flagella. And if you break this in a mutant, the synchronization goes away. Okay, so there has been this doubt that hydrodynamic alone is not sufficient to explain this synchronization, and these physical connections between these flagella also play a very important role. Okay, so that's where we started our experiment. So we conjectured that, okay, let us just throw out the hydrodynamics completely out of the picture, and let us just look at the role of physical coupling. Okay, and let us go one more step further. Let us create a dry macroscopic system which exhibits run and tumble motion. Okay, so just to give comparison between, okay, this is our system. So alga here, it has two components which are flagella, like I said, and their main role is just to propel the body of the organism forward. And it is in a low Reynolds number environment, so inertia does not play any role. And synchronization mechanism can also be a physical connection. Therefore, motivated by this, inspired by this, we created our robotic analog, which is these two robots, which I'll talk about. They're connected with a rigid rod, which kind of models the distal fiber I talked about. And these robots are active Brownian particles. They have an overdamped active motion, and the rigid rod is a source of synchronization in their orientation. And let us see whether that works. So this is what our robot is. It's a programmable robot, quite big. This is 2 rupee coin for the comparison. It has two wheels which are motorized, and they're connected to the motor driver and motor through these mechanical gears, right? And it responds instantaneously to the microcontroller signal. So therefore, we can model this as a completely overdamped macroscopic object, which is quite unique to this system. So this is how they move. So we can actually quite accurately sweep through the various values of rotational diffusion constant. So this is an active Brownian motion at different dr values, and this is how we connect them. So they are connected at these pivot points, which are at a distance delta away. And there are many parameters that we can change, but they are kept at mirror symmetric points around these robots. The idea is that this active force will create a left-handed torque on one robot and right-handed torque on the other robot. But since they also have their stochastic noise that will either cancel out this torque or act in the same direction. And this will stochastically run and tumble. So that was our entire hunch. And let us see whether that works. And of course, we can vary all these parameters, which we have done. And when we set them in motion, this is what happened, right? So this was basically the title slide movie. So they indeed show run and tumble motion. I'll take one more minute. So this is basically the main result of the paper. So you can see they run, and once in a while they tumble. These are the parameters. And we are here plotting the pair angle. So every time they are running, their orientations are aligned, call it synchronized. And every time their orientations are misaligned, it goes through this asynchronous cycle. And of course, we quantify this. Again, the run phase corresponds to a very high speed, and tumble phase corresponds to a very slow speed, as expected. And we can also, based upon this, take very long videos and characterize all the run times and tumble durations, etc. And encouragingly, the run times are exponentially distributed, as seen in both E. coli as well as Chlamydomonas systems. And even the tumble durations are, the average runtime is 5 times greater than the tumble duration, which is also true, at least for the E. coli system. And one important point, and after that I'll end. Inertia, like I said, our system is overdamped, but here we artificially created underdamped system by just spilling coconut oil on the glass slide. So this dirty stains that you see, this is just coconut oil. And as a result, the robot shows a lot of slipping, right? So which basically adds the inertial time, finite inertial time in the system. And we see that these tumble events, they tend to disappear, right? So there is no sharp turn anymore. It is just a uniform run event. So therefore, like overdamped limit is really important for run and tumble motion to emerge, which is quite obvious because they all live in very low Reynolds number environments, but we can demonstrate that, which is, I think, a neat thing. So I'll just skip all these details. There is a very nice phase, rich phase diagram. So we can quantify the tumbling rates, and there is a lot of interesting features, which obviously I won't be able to go through. And with our collaborators, we also simulate our system. And again, we throw out the inertia part, okay, which is very important. And the experiments and simulation, they qualitatively match with each other. And even the other statistical features, they also show very similar results. I'll skip all this. We understand the features of the phase diagram. We compare with theory results and experiments. I'll skip all that. And I'm happy to take questions. Thank you. Yes.
What is the underlying dynamics of each of the robots? What's active Brownian?
They are over-damped active Brownian particles.
Oh,
yes.
So I was wondering, like, are there any physical reason why this would be, both of the robots should be coupled? I mean, run for some duration and then tumble, like.
Okay, so the basic picture is quite clear from here itself. So you can see that by design, we have designed it in such a way that the force due to the active force will cause a torque on this robot, which is the propulsion force multiplied by this separation delta. But again, there is another rotational noise on this robot. Okay, so when the rotational noise is acting in the wrong direction as torque, it will continue to move in one direction. But when stochastically, once in a while, the noise will act in the same direction as torque, and the robot will turn and it will undergo a tumble duration. So that is how we introduce tumbling in the system. So run and tumble essentially is a combination of translational and rotational dynamics, which occur stochastically. And of course, we can vary the parameters like rotational, the diffusion constant, and we sweep through entire phase space of that. Yes. Huh.
Just curiosity. So these were miniature versions of the Firebird or the spherical robots?
Sorry, say that again.
These were the miniature versions of the Firebird or spherical robots?
No, these are quite big robots.
No, I know that, but that Syscon has developed these miniaturized Firebird robots.
Okay,
are you using those, or you reassemble them from scratch?
Yeah, I'm not aware of those robots.
Yeah. But these are not spherical robots, right?
No, these are circular. They are like disc shaped. Yes, they are not spherical.
So in real system, two flagella means synchronization into flagella comes from hydrodynamics, or?
That is a big debate in the community. That is what the understanding was for many years, that it is hydrodynamics, but then they created a mutant where they removed that distal fiber connection and the synchronization failed. So they thought that, I think the current understanding or the consensus is it's combination of both. But we show that like even in dry system, okay, I'm not saying this is a chlamydomonas, but we are inspired by those kind of results, and we are just working with the physical connection. There's no hydrodynamics here.
Okay, so if no more questions, let's thank the speaker again.